In a previous lesson, we went through the external arterial bloodline. Now we will explore the heart of the dialysis machine, the dialyzer. I've got something special for you prepared, but first, let's start with the basics in our learning matrices. You can imagine the dialyzer as an artificial kidney in its function, but not in size and shape. It is designed as a plastic cylinder filled with hollow, semi-permeable fibers. The dialyzer has 2 connections for blood circulation and 2 connections for dialysate circulation. The purpose of the dialyzer is to remove excess water and wastes from a patient's blood, once the kidneys can no longer handle the process. Let's look inside. Blood enters the dialyzer through the arterial header. Inside the header is a clay-like potting material that holds thousands of hair-thin hollow fibers in place and keeps them open so blood can flow through them. Dialysate flows around the outsides of these fibers. With each pass through the dialyzer, blood leaving the fibers through the venous header has been cleansed of some of the impurities that would normally be removed by the kidneys. Let me show you the dialyzer from a different angle. Once again, hollow fibers inside are held together by the potting material. Blood flows through the insides of the fibers, while dialysate flows around the outsides. Does this concept make sense?
I think I got it.
This is an important point, and I'm glad to know you understand. Let's continue. As I said at the beginning, I have something special for you. Please touch the dialyzer connected to the machine. Can you hear me?
Loud and clear.
Excellent. You are now inside the dialyzer, safely contained within this small living unit. What you see all around you are the hollow fibers. They are small, 200 microns in diameter, and the wall thickness is just 40 microns. By comparison, a human hair is about 75 microns in diameter. Pores or holes are randomly distributed along the surface of these fibers and vary somewhat in size. The dialyzer contains thousands of these fibers. I showed you earlier in the clinic that blood flows through the insides of these hollow fibers. It is noteworthy that in this setup, the blood flows from top to bottom. In other dialysis machines, the flow can be in the opposite direction, from bottom to top.
What are we removing from the blood?
During dialysis, we aim to remove excess water and solutes, urea, creatinine, beta-2 microglobulin and electrolytes, such as potassium, sodium, magnesium, and phosphorus. Albumin, which is a protein that the patient's body needs, is too large to fit through the dialyzer pores. The pore size was determined on purpose, so dialysis will not remove albumin or red blood cells. As we mentioned earlier in the clinic, dialysate is a prescribed mixture of purified water, acid, a bicarbonate buffer, and electrolytes. Excess water and wastes pass through the pores of the dialyzer and into the dialysate. The nephrologist can tailor the dialysate to meet a patient's needs. For example, when there is less potassium in the dialysate, more will be removed from the patient's blood, which can allow a patient to eat more healthy fruits and vegetables. Dialysate flows countercurrent to the blood. This means in the opposite direction.
So, the patient has water and wastes in the blood that are normally removed by the kidneys. How do the hollow fibers and dialysate work to remove them?
Great question. The principles of diffusion and osmosis play out inside the dialyzer, and these are how wastes and excess water are removed. Diffusion is movement of a solute through a semi-permeable membrane from a high concentration to a lower one. For example, urea diffuses through the hollow fibers from the blood into the dialysate. Diffusion will go on until both solutions have the same concentration. In dialysis, we create a concentration gradient on purpose, so we can make use of diffusion. Dialysate does not include urea or other wastes. Waste solutes that are small enough, such as urea and creatinine, pass through the membrane pores will move out of the blood and into the dialysate. All molecules, and especially larger molecules, are helped through the membrane by the flow of fluid that flows through the membrane from the blood compartment to the dialysate compartment. The larger molecules can only go through larger pores. This is convection and adds to waste removal for solutes like beta-2 microglobulin. Osmosis is like diffusion, except that water moves through a semi-permeable membrane to equalize the water levels of two solutions. In this case, the two solutions are blood and dialysate. Osmosis alone is not enough to remove all of the excess water for most patients, so we add a pump to pull water out of the blood. This is called ultrafiltration. The dialysate solution contains specific electrolyte, such as magnesium, potassium, calcium, because they are essential to the body's function, and removing or adding too much of them would be detrimental. These solutes will diffuse across the membrane in either direction, depending on the level in the patient's bloodstream and the level in the dialysate. In most cases, the level of these are higher in the patient's bloodstream, and will equilibrate to a level halfway between the blood and dialysate. I hope that seeing diffusion and osmosis has helped you to understand it better. Next time we are together, we will discuss the last part of the extracorporeal circuit, the arteriovenous bloodline. Thank you for your attention.